Welcome back. Today we are talking about energy and enzymes. This leads into talking about the cellular pathways that are used for energy transformations inside cells, which will be the next three lectures when we talk about cellular respiration, photosynthesis, and fermentation, those three pathways. So just to get us started, what is energy? Energy is the ability to do work, and in terms of cells, we are talking about life work. We are talking about movement work or transport work where we're opening gates or closing gates. This kind of energy is also needed to build different chemicals up inside or break them down. The international unit of energy is a calorie, and there are all different kinds of energies in our world. There's potential energy, energy of position, kinetic energy, energy of motion. There's also heat and chemical energy and chemical bonds, mechanical energy. So lots of different kinds of energy inside of our world. There are two big laws that govern how energy behaves in and around cells, and they govern not only cell energy, but also the energy of the world around us. So the first one is known as the law of conservation of energy, or the first law of thermodynamics. It states that energy cannot be created or destroyed, but it can be converted from one form to another. So if we see this cheetah, like in a living thing, it is eating chemical energy in its prey item, and then transferring that or converting that chemical energy into energy of motion or heat energy as it heats up its body, these kinds of activities. So this is the first law of thermodynamics. Energy cannot be created or destroyed, but can be converted from one form to another. The second law of thermodynamics states that with every energy transfer, like our cheetah transferring chemical energy into energy of motion or heat, with every energy transfer or transformation, they don't transfer 100% of all the energy. So they aren't 100% efficient. Some energy is lost at each of those transfers. You knew this because of our food chain and food web and energy diagram sort of lesson that we learned in ecology. We said that as energy moves through our ecosystems, some is lost at each of these transfer steps. So energy has to be like sort of replenished frequently to our ecosystems in order to sustain them. And the same thing happens with us here on planet Earth and our cells. In order to make them work, we are going to do these energy transformations, but we will need energy coming in and being replenished so that we can sort of start these cycles over and over again. I show a diver over here. If you climb and change your potential energy and then dive off a platform, you change some of your energy of position into energy of motion and heat as you're diving down. Some of that energy cannot be claimed back to you. So in order to dive again, you have to climb the steps again, right? And change your position so that you can dive a second time. Now, what kind of energy does a cell use? Cells have a special form of energy that is very easy for cells to break down and build up, and it is called adenosine triphosphate, or ATP. Adenosine triphosphate, I hope you notice over here, has a ribose sugar, an adenine nitrogen base, and some phosphate groups. And it makes almost this letter L repeating unit. And I hope you recognize that as a nucleic acid. So this is something that belongs to something that's related to RNA, DNA, so those kinds of nucleic acids. The difference between this particular structure, ATP, and an RNA would be a couple of extra phosphates on the end here. So you have triphosphate, or three phosphates that are negatively charged, that are stacked quite closely and crowded together. Now, because of the law of opposite charges attracting one another, when we get the same charge next to each other, it's like you and your siblings in the back seat of a very small car. You don't like it, right? These chemicals don't like to be stacked so nice and neat with the same charge one after the other. And so what's interesting about these phosphate groups is we can break off a phosphate, and when we break this, it actually releases energy. So we can move from ATP, triphosphates, to ADP, which is adenosine adenosine diphosphate, two phosphates, by breaking one off and releasing energy. We could even move from ADP to AMP, adenosine monophosphate, and break off until that lasts another phosphate group and release some more energy. And so this particular molecule is quite nice. It's poised and ready to sort of be easily sort of broken apart to release energy. And then we can also put it back together to replace that energy for future use. Almost like a rubber band that you can pull and stretch over and over again. And it does some work for us, right? So it does some living work for us. Now, what is the ATP story like for cells? In your cells, ATP is actually built and broken down over and over and over again. In reality, a working muscle cell, so if you were running a marathon and working a muscle cell, you are spending about 10 million ATP and then regenerating 10 million ATP per second. So you're doing this story over and over again. When we go from ATP to ADP, we release energy so we can do our energy of motion, transport energy, build things up, break things down. This is your energy of your cellular life work that you do. So when we break down ATP, we release energy for that cellular work. But we can also use this process called cellular respiration to put this molecule back together. We can transfer the chemical energy harvested from the foods that we eat back to this molecule and reset it, almost like a rubber band where we're pulling it back again so that we can release it and do some work. The same thing is happening in your cells. We can use the food and transfer the chemical energy in the foods that we're eating, breaking those bonds and transferring it back to this molecule so that it goes from ADP back to reset with that third phosphate group or adenosine triphosphate back to its original form. Your cells, again, in a working cell is doing this millions and millions of times per second, this particular cycle. Now, what kind of molecules facilitate this ATP story or all the chemical sort of reactions that are happening inside your cells? I kind of think of it like a square dance where it's like swing your partner round and round. It's like your chemicals have to interact, and sometimes those interactions have to be facilitated or enhanced, right, or encouraged. And so the chemicals that do these kinds of facilitations are called enzymes. Enzymes are proteins. They're going to make the ATP story happen. A full suite of sort of enzymes work to make cellular respiration happen. And the same thing with fermentation and photosynthesis. There's lots of things going on to make those energy reactions happen, and many enzymes working together. But enzymes are mostly proteins, and they help us speed up our chemical reactions and facilitate them inside our body so that they happen in a timely fashion. Now, where do enzymes come from? They are actually proteins. And so most enzymes will be created via DNA. So you'll have a code for a particular enzyme, and then DNA will actually send out messages to make enzymes so that you can facilitate your chemical reactions. Again, enzymes are proteins that speed up chemical reactions. So how do they do this? How do they speed up a chemical reaction? Enzymes really do one important thing. Enzymes lower activation energy of reactions, and by lowering activation energy, it causes these reactions to go faster. Now, activation energy is the energy required in a reaction to make it go forward. It's almost like this hump in the hill. There's this wall, this activation energy that a particular set of chemicals might need to get over in order to cause a reaction. In this case, we're breaking apart this reactant into two different products. What an enzyme does is it basically sits on that wall and lowers that wall. It lowers the bar of energy that is necessary in order to see a reaction happen. Think of a match that is unlit. You can hold that match for 100 years and it would never light, right? That activation energy, there's a bar in place that is holding that match together, making it not light. However, if we lower the activation energy by just rubbing it and applying some friction to the head of the match, it's enough heat and it's enough friction to start that match going. And once it's going, then that reaction can go all the way to completion. It can burn the whole match, everything that is around it, right? Until there's nothing left to burn. Enzymes cannot, I hope you notice, they cannot change the amount of reactants, because that's what you're going to be coming in to the body with, or coming into a reaction with. That's going to be external forces typically that are providing those reactants. So enzymes don't change the amount of reactants, and then they really can't change the products, because that's based on how much of the reactant you have. So all enzymes can do is lower that activation energy and then make a reaction go faster. There are different parts of an enzyme. So over here, I'm showing the enzyme sucrase, which breaks down table sugar or sucrose. You'll notice that a lot of enzymes have that ASE ending with them, and that's how they are named. This purple piece is the actual protein body that holds an active site. And active sites are regions that can fit and dock a specific chemical. And it's almost like lock and key style. They will only fit one kind of chemical and do one kind of reaction. So in the case of the enzyme sucrase, we are breaking sucrose, a disaccharide, into its two monosaccharide pieces, glucose and fructose. So we are splitting this disaccharide into two pieces. And so this active site will bind the substrate, and the substrate is what participates in the reaction. It actually is changed by the end of the reaction. So I hope you notice the enzyme is not changed. It is actually a catalyst. It facilitates a reaction, helps stimulate a reaction, but doesn't get changed itself by the reaction. This means inside of your body, you can have a small amount of enzyme, and then they'll do the reaction over and over again for a lot of output. Think of like your stomach. You don't need a lot of enzymes in there. You just need some so that you can break down the foods that are passing by. And so they can actually, as the food sits in your stomach, these enzymes will like just continually sort of change proteins into their amino acids, and they can do a large number of reactions over and over again and be recycled for that. When the substrate is docked on the active site, the enzyme actually folds over it to hold it more closely. This is going to help it lower the activation energy, and it's almost, we call it induced fit, but it's almost like a handshake. When the enzyme and the substrate come together, they don't just like pass by each other like woo, but they actually like do almost like a handshake where they squeeze one another. And this induced fit allows the enzyme to hold that substrate quite closely. Now, how does this induced fit speed up a chemical reaction? When the enzyme is holding that substrate very, very closely, it can be adding and subtracting charges, right, that make a particular chemical react faster. It can also bring reactants and into a certain orientation so that they're more likely or closer together so that they can react more easily. Or sometimes even the squeezing, just applying pressure, encourages the reaction to happen faster. So this induced fit helps lower the activation energy and speed up these reactions by sort of squeezing and adding and subtracting like charges, changing chemistry, bringing things closer together so that reactions can occur. Now, can enzymes be stopped? Yes, right? We need to be able to stop them so that they don't go too far. Chemicals that stop enzymes from working are called inhibitors. And inhibitors come in various types. I'm going to walk you through competitive and non-competitive, but there are some other types like allosteric enzyme systems, etc. I showed this danger poison sign because a lot of enzymes are stopped, right? There are certain enzymes that stop working in the presence of certain poisons. And so a lot of the reason why poisons kill you is because they're inhibiting or blocking an enzyme from functioning inside of your body. So how do inhibitors work? In competitive inhibition, the inhibitor is going to mimic the substrate in size, in chemistry, in the way that it like sort of fits in that like sort of active site. And the inhibitor is going to compete for the active site and block the substrate from being able to bind. And if you block the substrate, you stop the reaction. I'm showing this a regular reaction with the substrate in A here. And then this is showing in B, competitive inhibition, where you have like two things competing for this active site. This competitive inhibitor, if it binds first, will block the substrate and then the reaction stops working. Penicillin actually works this way in order to kill bacteria, it actually inhibits an enzyme necessary for building a cell wall around the bacteria. And so by inhibiting that, the bacteria can't reproduce and build more bacteria, and so you can kill the bacteria in and around your body. Viagra is another competitive inhibitor. It works to stop an enzyme so that you get blood flow to the penis and you get an erection, right? So a couple of different sort of common examples that you've probably heard of that are actually competitive inhibitors. Non-competitive inhibition is shown in C over here. This is where your inhibitor binds at a distance and then changes the active site's chemistry or size or shape so that the substrate no longer can fit. If you can change that active site and block the substrate from being able to dock, then you can stop the reaction. There are lots of poisons that fit into this class as well, like sarin is a common terrace bomb that sort of decreases the way that the muscles and the lungs like inflate and deflate so it can stop breathing, and then people die. And strychnine influences like nervous and muscle control, so that's often a common poison that's used in baiting sort of animals and various things like that. So these are working because they are blocking different nervous system and muscle sort of enzymes that are necessary for the function of how we like sort of move and respond in our environments. Are there other ways to stop enzymes? And the answer is yes. There are other kinds of inhibition systems like allosteric enzymes often have many different kinds of inhibition sites and then even some promoter sites. A lot of the human enzymes work this way so that you get a range of functionality. You get some speeds that are very, very fast when you need something very fast, and then medium speeds, and then even like to the point of like stopping an enzyme altogether. So you get this spectrum of almost like speeds for working your enzymes. Enzymes also require extremely specific conditions, like temperatures have to be just so, and pH and chemical concentrations. Otherwise, your enzymes can denature or fall apart like this picture is showing. Basically, the take home is if you mess with an enzyme's tolerance levels, it often will stop working altogether. So that's all I want to say about an introduction for energy and enzymes. I will see you next time.